All right, so welcome back to week 4. So today we are just actually only going to be talking about pop health because last week covered bacterial pathogenesis, so micro, quite well. So if you want to look at that content, probably just go back to the PSP last week. Okay, so in pop health, we're going to be talking about study design. Study design is a really important topic in pop health because in your assignment, you're going to be asked like, what is the study design of the thing that you're doing? And also in your exam, you can get a few questions about it as well. So make sure you know key features of each study design and how you would kind of be able to identify what a study that you've been given is. Okay, so we have a few different study designs, but we'll break it down into the groups that they're in and what key characteristics they have. So if we look on this picture, you can see that one of the big things is, did the investigator or the researcher assign exposure? So exposure is pretty much like some factor that can change your likelihood of getting a disease or not. For example, smoking when we're talking about lung cancer. So here, if the researcher has not assigned the exposure, that's called an observational study. And then if there's a comparison group, that will become an analytical study. And if there's no comparison group, it'll be descriptive. And we'll talk about what specific studies come under each topic in a bit. And then if the investigator has assigned exposure, that is called an experimental study. And there's 3 different types, but the main one that you need to know is the randomized controlled trial. So that is when the sample or the participants have been randomly allocated into a control or an intervention group. So if we look at this pyramid it's pretty easy to remember, and I would suggest remembering this order. So you have the case series and case report, which would be the observational descriptive studies. So where there's no exposure assigned and there's no comparison group. Those are kind of at the bottom, as well as ecological studies. Then the next 3, which are the green ones, these are the analytical studies. So they get a little bit more reliable, a little bit less external factors influencing your results. And then you have your RCTs. So your RCTs are your ones here where we've assigned our exposure and we've also got random allocation. And then at the very top, we have meta-analyses. I'm not going to talk much about them in this thing, but pretty much a meta-analysis is where we get a bunch of different studies, usually of the same study design. Obviously, the most reliable would be if we got like a bunch of RCTs, but sometimes you can get a bunch of case control, a bunch of cohort studies, and you collate all of the data from all of those studies to get this like big picture idea. And what do I mean by top of the pyramid and bottom of the pyramid? Pretty much what I'm saying is the ones at the top are just of better quality. And we'll talk about certain biases that can affect the quality of a study, but we just have less external influences affecting the results for studies up the top versus studies down the bottom. On the top left, I've just got some keywords that you should be familiar with when we talk about study designs and be able to identify in a study what each of these would be. So a case, that is the person who, if someone has a disease or an event of interest, control is the opposite, so someone who doesn't have that. And then we have the exposure and the outcome. So the exposure is a factor that would change our likelihood of an outcome occurring. An outcome is kind of in a lot of your medical studies and medical research stuff, it's development of a specific disease. Okay, so firstly, we're going to talk about observational descriptive studies. So what does that mean? Observational is where we are not assigning our exposure, and descriptive, which comes under observational, that means we have no comparison group. So we have 2 types. We have case studies or case series as well, and then ecological studies. So a case study or case series, this is pretty much where so case study, is where we have a single patient. Case series is where we have more than one. So this is pretty much like in a hospital, if something quite unique happens or you notice a specific condition in a patient, you might write up a report about them, maybe in multiple patients. But you can see how that is not fully reliable. There's not many people that you're studying, and there's just a lot of biases that can come into play. Now, under each study, I've written advantages and disadvantages. I'm not going to go into them in this video, and it's probably more helpful if you look at them yourself. But I'm going to talk about the features of each study design, and you can kind of infer most of the time what is advantageous about it and what's not from the description. So with an ecological study, we are using the unit of analysis is a population and not an individual. So what I mean by that is we might look at Australia as a whole or Canada as a whole, as opposed to the individuals themselves. And this kind of allows us to either look at different populations at the same period of time. So we can look at Canada and Australia now in 2020 and how many people in each country smoke. Or we can look at the same population at different periods of time. So how many people in Australia smoked in 2010 and how many people in Australia smoke in 2022. One key disadvantage of an ecological study, which I will touch on, is the ecological fallacy. And this is pretty much where we might attribute the characteristics of the group to the individuals. So pretty much if we say on average, Australians drink 20 glasses of wine a night. We could under, like, if we were going by the ecological fallacy, we would then attribute to like an individual Australian, okay, they drink 20 glasses of wine a night, which might not be true, because they might be having 1, 0, they might be having 100, who knows. But that's obviously, you can see why attributing the characteristics of the entire group cannot always be accurate. So then the next one is an observational study that is analytical. So we're still under the observational, so we're not assigning that exposure. And then an analytical study is where we do have a control group, and we'll see how those control groups come about as well. So we have cross-sectional, cohort, and case control studies. So a cross-sectional study, this is a prevalence study. Now, a lot of the time, if you see prevalence in like the question stem, it can really clue you to the fact that it's a cross-sectional study. Obviously, make sure you read the whole thing and still take into account other things. But yeah, it's a really key feature because you're looking at the exposure and outcomes at one particular time. So in 2020, how many people smoke? And how many people have lung cancer, for example. A big disadvantage of this is that we can't determine the direction of this relationship. So because we're taking it at one specific point of time, we can't make any conclusions about whether the smoking is what causing the lung cancer, or whether it's the lung cancer that's causing the number of people smoking. So that's not a relationship we can draw because it's at one specific time. Whereas in a case control study, and we'll talk about cohort studies in the next one, you can kind of start to establish some kind of, this is the exposure, this is the outcome, because we're looking at an exposure that's occurred sometime before the outcome. So in a case control study, this is a retrospective study, and that's a really key word when it comes to case control, is that I'll now talk about what retrospective means in a second, but if you see this word, it can really clue you in on what type of study it is. And I'll quickly just go to the next slide. In cohort studies, we have prospective and retrospective cohort studies. So just because you see the word retrospective doesn't mean it is, but pretty much what a case control study is we are going to start with the outcome, so lung cancer. And then we're going to look back in time to find the presence of an exposure, so smoking. So in 2022, I found someone who has lung cancer, and now I'm going to investigate whether or not they were exposed, they smoked in the past, which is being exposed. So this can be one specific disadvantage of this is something called recall bias, which is pretty much where someone who has lung cancer might be more or less likely, would actually be more likely to report that they had smoked in the past versus someone who doesn't have lung cancer. They might be less likely to remember whether they smoked or not because it's just not significant to them. And another thing, obviously, is the fact that we're getting data from the past. So that's going to impact your results because the data might not be reliable. You, as a researcher, do not have control over the quality or how the data is being taken. Now, in a cohort study, so in the last one, this is a very important distinction. So case control, we're looking back in time. So we're starting with the disease and we're going back in time to find the exposure. In a cohort study, we are starting with the exposure and we're going into the future to get the outcome. And that's a really key distinction, and it might help you become less confused when we talk about a prospective and retrospective cohort study. So prospective is what I just explained. So I'm going to find someone who smokes, and then I'm going to follow them into the future to see whether they get lung cancer or whether they don't get lung cancer. Retrospective, we're still starting with the exposure and we're going to follow them into the future to see if they have the outcome. Where the retrospective kind of word comes into play is because we're going to find a person. We are then going to use maybe past medical records or information data that was collected in the past to see have they smoked. But then from this point in time, we're still going to follow them into the future and see are they going to develop the disease or not. So at the moment when you start the study, it's not like they have the outcome yet or they don't. So in terms of what a cohort study needs to, the different features of it needs to be accessible, of course, and the participant can't have the outcome at the beginning of that experiment because you're going to investigate whether or not they will develop it. And then also this idea of a follow-up. So we'll talk about this in RCT as well. In cohort and RCT trials, you have a follow-up period with your participants. So every few months or every 5 years, you have to meet up with your participants, see how they're going, see if they've developed the outcome, see if there's any adverse effects, etc. So this can often be a disadvantage in a study. Firstly, it's expensive, but also it can mean that a lot of participants drop out, and that will obviously affect your studies because you won't be able to use that data. And in a few weeks, you might talk about how that's addressed with something called intention to treat analysis. So there's specific outcomes. So how do we quantify the results that we get from a case control or cohort study and also RCT study? So we have these things called an odds ratio and a relative risk. An odds ratio we will use for case control studies, and a relative risk we use for cohort studies and RCTs. So that's really important to remember. Don't use the wrong value for the wrong study. So an odds ratio will estimate any relationship that is there between the exposure and the disease. And that's calculated this way. So you can see how we put the odds that a case was exposed. So this kind of ratio of A over C. So you can see A is a case, so someone with the disease who was exposed over someone with the disease who wasn't. And then we put that value over B and D. So, and then just some high school maths for you there. And then we have relative risk. So the risk of the outcome, we're looking at the risk of the outcome happening in someone who is exposed versus unexposed. So this pretty much here is how you calculate it, and you can take note and kind of remember that formula as well. It might also be useful just to kind of understand what these numbers mean and why. So here, so A over C, we're looking at the ratio and the risk that's here, and we're dividing it by the odds of people without the disease being exposed or not. And that can help you kind of avoid the whole memorizing thing. So then in terms of interpreting the OR and RR, this is the most important part of these. So if it's less than 1, it means that the risk of the event happening or someone developing the disease in the intervention group is lower than in the control group. If it's 1, it means the chances are quite equal. And then if it's more than 1, the risk of developing the disease in the intervention group is more than in the control group. This could mean that, say, like if it's less than 1, the intervention might be preventative. And if it's more than 1, the intervention might be like causing or more harmful. But obviously keep in mind the fact that there are potential biases that can, and external influences that might make that causal relationship a bit less unclear. So you can't always say it's because of the exposure, but it can give you a bit of an indication of whether it makes something more or less likely. Okay, so the last one is experimental studies. So these are when we actually assign the exposure. So there's 3 different types. The one that you pretty much only have to look at is randomized control trial. So a randomized control trial, this is the gold standard of epidemiological study, and that's because we control so many external factors and a lot of those biases that often skew the results in the other studies that we just talked about. So this is really important. So we have the population, we take a sample, and a key feature of a RCT is that we randomize how we've allocated the sample into either the intervention or control group. And then we just calculate who in the intervention group got the outcome, who didn't, and then who, and then the same thing pretty much in the control group. So 2 key features of an RCT is blinding and random allocation. So blinding is pretty much when we make certain groups know whether or not a participant is in the intervention group or in the control group. So there's 3 different types. There's an open RCT, a single blinded, and a double blinded. So in an open RCT, everyone involved, so the researchers and the participants, know which intervention is given to each patient. So you can imagine how that could influence results because people have their own biases and their own ways of maybe reacting to the fact that they know they have a control or that they know they have a specific intervention. Then we have a single blinded one. So in a single blinded one, we have one group of individuals, which will usually in this case be the participants. They will not know the identity of the intervention that is given to them. So the researchers in this case are usually aware, but the participants don't know if they have the placebo or if they have, say, the drug, for example. And then in double blinded, we are going to have both of those, so both the researcher and participants not know who is given the intervention and who is given the placebo or who is in which group. And this is really this is the best way pretty much to do it and will limit the most external factors impacting your results, because on one hand, if the participants don't know, they're not going to act differently or kind of have those psychological internal influences. And then on the other hand, you have the researchers who will not, if they know a certain participant has an intervention or if they have a placebo, they might collect data differently. They might put more significance to certain adverse effects happening, or just there's more biases that happens in terms of data collection as well as analyzing that data. And then random allocation. So this is a really key feature of RCT, and it's called a randomized control trial for that reason. And this is really important because we're minimizing the impact of baseline characteristics. So that's things like age or gender and sex. We're minimizing the impact or the differences on each group's results. So we're not going to have, one whole group of males, which is the intervention group, and then one whole group of females, which is the control group, because you can see how that would affect the result. One way to eliminate biases even more is randomly allocating through a process of allocation concealment. So that is when that allocating sequence is kind of hidden from the researchers who are actually assigning participants. So if the researchers themselves do not know anything about the participant and they don't know which group they're putting them into, it eliminates any biases that the allocators that themselves might have when they're assigning a participant to either group. So this is important. If a participant seems, a bit more sick, or certain characteristics that the researcher attributes to them, they might be more or less likely to put them in the intervention or control group, depending on which way favors them more. So if we just eliminate that from happening, it'll reduce selection bias. By the way, all of these biases that I've talked about, I've put it in the notes at the bottom of each slide. So if you're not really sure what those biases are, you can just have a read. And these are from your lecture, these two. So they're just helpful in terms of kind of summarizing main key features of each study design. But yeah, that's pretty much all. You can have a look at that in your own time. And that's pretty much all for this week. So if you have any questions, you can just let us know. And hopefully we'll see you back next week. Okay.